I'm going to say some things about complexity and how I think biology handles complexity. I'm going to say a few things about emergence and some kind of fairly unconventional and possibly unpopular things about ways to think about this. We'll see towards the end. Generally, my lab is three-quarters wet lab biology and about a quarter computational people. So we straddle this interface between computing and the life sciences. And you can find all of the data sets, the software, the primary papers, everything is downloadable at our lab website here. And this is my personal blog where I write about what I think some of these things mean. So what I'm going to try to convey to you today are basically three things that we really need to get beyond neuromorphic architectures in our attempts to understand how intelligence can handle complexity. First of all, what I'll do is I'll show you some amazing and unconventional, in the sense that you wouldn't have seen these in standard introductions to biology, examples of intelligence. And I think what it's telling us is that life uses intelligence all the way down to the lowest scale, or the lowest level of organization. I'm going to try to highlight a few of the lessons that we've learned from all this, and in particular, evolutionary and mechanistic aspects of how we communicate with, not micromanage, the multiscale agential material of life. I think the complexity of the living organism makes it basically impossible to micromanage in the same way that we engineer with other types of materials. But that's okay. That's not how life regulates itself, and we can do better, and we can do what life does. And I'll show you what that is. And then towards the end, I'm going to say some fairly speculative things about the origin of embodied intelligence, and I think what it means both for biomedicine and also for AI and things of that nature. One of the key things that I try to do in our work is to develop a framework that allows us to recognize, create, and relate to really unconventional intelligences. And I'd like to be able to understand what they all have in common, regardless of their composition or their origin story. I seek unifying principles around familiar creatures such as primates and birds and maybe an octopus or a whale or something, but also very weird creatures, colonial organisms, swarms, cells, tissues, synthetic life forms, of which I'll show you some, AIs, whether software or robotic, and perhaps someday exobiological agents, so alien life maybe, and then also some very strange things that I probably won't have time to talk about today that aren't physical beings at all. And what I'd like to do is to develop frameworks that are not just philosophy. They need to move experimental work forward, and they need to show how specific philosophical ways of looking at these things actually lead to progress, both in biomedicine, which is a lot of what we do, but also in bioengineering, in computer science, and very importantly, in better ethics. I'm not the first person to try for something like this. Of course, this is Wiener al.'s work in 1943, where they tried to show the various steps in terms of cybernetics of how you get from passive matter to human level metacognition and so on. And the details of my work is on this stuff is here in this fairly lengthy paper. So let's talk about some of the unique features of the biological substrate, and I think this is important in establishing how far away from brains we need to go to really understand what's going on. The first thing I want to say is that biology kind of presents currently, and it has for a long time, presents a face to the outside world of the dominance of genetics. And the idea is that the molecular biology of the genome should really tell us everything we need to know. And the promise is, of course, that once we really tame the genetic information, we'll be able to do whatever we need to do. So I just want to explain how things are actually, I think, quite different. So this is a baby axolotl, and axolotls are amphibians that have little tiny forelegs in this early stage. This is a tadpole of the frog, Xenopus laevis, which we use all the time. They do not have legs in the early stages. In my group, we make something called a frogolotl, where we basically combine cells from axolotls and frogs. And now I could pose the following question: you've got the axolotl genome; it's been sequenced. We know we can read it. It's been annotated. We have the frog genome. We know all the genetic information from this species. Simple question: if I make a frogolotl, are frogolotls going to have legs or not? And if they do have legs, are those legs going to be made of just frogolotl cells, or in fact will they incorporate some Xenopus laevis cells? So the important thing to note is that we currently have absolutely no way of deriving that kind of information from the genetics. while the genetics specify the hardware that all of these creatures get, that all of the cells in these creatures get to have, so all the protein level, nano level hardware. What they don't tell you is the actual software which drives outcomes. In other words, the computations and the decision making that happens at different scales once that hardware is powered up. So we have to understand right now that knowing the specifications of the hardware, for example, in terms of the genetics, is not sufficient. Then you find out that actually these forms, while they're pretty reliable by default, you get these kinds of complex forms. There's actually incredible plasticity that's there. So here's a tadpole of that same frog. Here is the mouth. Here are the nostrils. Here's the brain, the spinal cord, and the gut is back here. What you'll notice we've done is we've prevented the primary eyes from forming, but we did put an eye on its tail. And this eye, we've traced it with red lineage label. You can see that it makes an optic nerve. The optic nerve does not go to the brain. It sometimes synapses on the spinal cord, sometimes on the gut, sometimes nowhere at all. And by using this machine that we built to automate the behavioral training of these animals using light cues, we found out that they can actually see perfectly well. Okay. Now, this is, and remember, they don't have any other eyes. This is their eye, does not connect to the brain. They can see, they can learn in visual tasks. So this is a puzzle, because typically you would think that a novel sensory motor architecture would need mutation, selection, evolution. You would need some adaptation to do this. Now, it works out of the box. The very first time you make these animals, never existed before. No problem. They can see the brain somehow interprets signals arriving from other pathways, folds it into their behavioral repertoire. So already we're starting to see that there's functional plasticity here that speaks against some kind of a hardwired system where the genetics just tells every cell where to be, and we know that's impossible anyway. We can go on and notice that this plasticity is actually much deeper than that. Here is a creature called the planarian flatworm. These little guys have many fascinating properties. Among them, they are highly regenerative. So if you cut them into pieces, each piece will regrow exactly what's missing and make a perfect little worm. But they're also smart. They can learn. So you can make. So this is what we did. We used a laser to etch little bumps into one region of a petri dish. We would feed them in this one region, and so that's called place conditioning. They would basically learn that this is where they should eat, and then we cut off their heads, containing their centralized brain. The tails sit there doing nothing, no behavior, until they grow back their brain. About 8 or 9 days later, they grow back their brain, behavior returns, and now you find out that they actually remember the original information. This was first actually discovered in the 60s by this guy named McConnell, but he did everything by hand, and he got a lot of flack for it because nobody thought that you could store information outside the brain. But we did it with this automated device that I just showed you, and he was right. It does work. And so what this tells you is that not only is there information stored outside the brain, but that information can be then imprinted onto the new brain as it develops. So behavioral information is actually moving through tissue, and we, there's many implications for this, but it's actually moving through tissue, being imprinted onto a new structure as it develops. Here's another example that I think is pretty cool. So this is a fly. And what this fly has is, and this is not AI or Photoshop. The first time I saw this, I thought this was definitely some kind of fake thing. What it's doing on its wings is running a stripped-down morphogenesis program in 2 dimensions for ants. So it's got these images of these other creatures on its wings, and it uses them to scare off predators and things like that because nobody wants to deal with the ants. But it's kind of remarkable, this degree of virtualization. Like you can run this simple morphogenetic program on the wings, but then there's a much more complex one, 3-dimensional program that is actually giving rise to the rest of the fly. And what we're starting to see here is really these kind of software aspects to the material where they don't just do the same thing every single time, but they have the ability to embody all kinds of interesting patterns of form and behavior, to the point where we can now see very specific context-sensitive, flexible error minimization skills. So for example, here's a tadpole. And this one has its eyes and its nostrils and so on. In order to become a frog, this tadpole has to rearrange its face. So the eyes have to move, the jaws, everything has to move. And it was thought that this was simply a hardwired process. Somehow, all the organs were told which direction to go and how much, and then you get from a normal tadpole to a normal frog. Well, we tested this because it's a strong claim of mine that the intelligence of these kinds of processes cannot be guessed or assumed from an armchair, that you have to do experiments. And sure enough, when we asked whether how much plasticity this has, what we did was we created the so-called Picasso tadpole. So everything is scrambled. The eye's on top of the head, the mouth is off to the side. It's like a Mr. Potato Head doll put together wrong, like everything is in the wrong place. And when you do this, you find out that they make perfectly normal frogs, because everything will actually move around in novel paths, sometimes actually moving too far and having to come back, until you get to a normal outcome. So this is very interesting. What the hardware actually gives you is not a rote set of movements. It specifies an error minimization scheme relative to a set point, and we'll talk in a minute about how that set point is encoded. But what it's giving you is a system that can start off in different configurations, unexpected, abnormal configurations, and still get the job done and get to the right location in anatomical morphospace. This, by the way, is one definition of intelligence by William James. It's the ability to reach the same goal by different means in some arbitrary problem space. So we now understand that this material that these things are made of is extremely flexible. It does all kinds of problem solving. As any problem-solving agent, however, it is also hackable, which has good news and bad news. Just to show you one example that I like from the plant world, this is an acorn. Acorns very reliably make oak leaves, and so you might think that, well, this is what the oak genome does. It encodes this particular kind of shape. Except that along comes a bioengineer, a non-human bioengineer, this little wasp, and what the parasite does is leave some prompts on this tissue that basically hack the morphogenetic competencies of the cells and cause them to build something completely different. So we would have no idea that these flat green things could build a complex gall structure like this, this like round red yellow spiky thing, or this, because under normal circumstances they always build this leaf. So it's clear that the material has all kinds of capabilities that is very hard to guess in advance. You have to do a lot of work, and we have a lot of ongoing projects in our lab to automate the process of discovery. Presumably it took millions of years for the wasp to be able to do this. We want to be able to do it quickly. And again, as I'm going to emphasize again and again, not by micromanaging the cells, not by genomic editing, not by telling individual cells what to do, but by high-level prompts. And the interesting thing about it is that it's kind of a two-way IQ test. In other words, the complexity of the thing you want the material to build is kind of dependent on our or the hacker's level of sophistication. So bacteria basically make these featureless blobs. Fungus doesn't do that much better. Nematodes, still kind of simplistic. But mites are starting to get better. By the time you get to insects, you get this beautiful structure that they can get the leaf to do. So we have to learn ourselves. We have to improve our own capabilities before we can get the material to do the cool things that we want it to do. So the last thing I want to show you is a little bit of a problem solving, another example of problem solving in a different space, this time not anatomical space, but a different space that is actually very relevant for robotics. So here's the experiment. So we have these flatworms. We put them in a solution of barium chloride. Now, barium is a non-specific potassium channel blocker. Blocks all the potassium channels. Most of the cells freak out, especially the cells in the head. The neurons are very dependent on potassium metabolism. Their heads explode. Over the next 18 hours or so, their heads literally explode. But if you leave them in the barium, in fact, refreshing the barium, what you will find is that fairly quickly they will build a new head, and the new head is completely barium adapted. The new head doesn't care about barium at all, no problem. And so we thought that was remarkable, and so we did some transcriptomics, meaning asking what genes do these barium adapted heads express that standard heads do not? What happened transcriptionally in terms of gene expression? How did these heads become barium adapted? And we found a couple things. First of all, it really is only about a dozen genes, and also individual worms all find the same solution to this problem. They all find the same gene expression. Now here's the amazing thing. just imagine you've got this physiological stressor that you've never seen before. Planaria are not typically exposed to barium in the wild, so there's no reason to think there's any kind of evolved response to this. You've got this incredible physiological stressor that's totally novel. You have a library of 20,000 effectors, roughly. Those are the genes that you could turn on and off. So tens of thousands of potential actions that you could take, meaning steps that you could take in transcriptional space. So it's a high-dimensional space. And it's a little bit akin to, Hans Moravec had this thing he visualized called the Moravec bush, which is a robot with a very high number of effectors. And he was asking the question, how do you design a controller for something with a huge number of effectors? We don't really have efficient algorithms for that. But this is what the system is doing. It's faced with a novel problem in one space, so physiological space. It has actions that it could take in transcriptional space. And in a very rapid order, it identifies exactly the dozen or so effector firings that it has to do to solve this problem. It's a remarkable example. There are many others. We could do this for hours. I could show you all kinds of wild examples. But this material is really good at improvising novel solutions to problems it hasn't seen before. So what we want to do now is talk for a little bit about how this actually works. Okay, and so here we go. I'm going to try to explain what's going on under the hood to the extent that we know. There are many open questions here. The first thing is that life really self-assembles from something that people would call just physics. So unfertilized oocyte, little bag of chemicals. People look at and they say, well, that's not a cognitive system. That's squarely within the province of biochemistry. That's just it's a machine. It follows mechanical rules. Well, fine, but slowly but surely that thing transforms itself into something like this, or even something like this, that's going to make claims about not being a machine and having true intelligence and all of that. The one thing that we definitely learned from developmental biology is that there is no magic lightning flash. There is no special time at which you click over from being a mechanical system into having a mind. This is a slow and gradual scaling process. And by the way, not the end of the line here, because there are some other processes that can still happen that are actually very, very interesting that I'll mention briefly. So what's actually happening here, and this is all described in this paper, is that by interactions that we are now beginning to understand, this material is slowly but surely enlarging its cognitive light cone. What I mean by cognitive light cone is the size in both space and time of the largest goals it can pursue. So not the reach of its effectors and sensors, but the size of the goals in some particular space, for example, in space-time, of the goals that it can pursue. And we are now starting to understand how that cognitive light cone is scaled up. But critical to understand here is that these cognitive light cones don't just project into the standard three-dimensional space of behavior. We as humans are completely obsessed with 3D space. We are pretty good at noticing intelligent behavior of medium-sized objects moving at medium speeds in three-dimensional space, and we call it behavior. And then we look at other things, such as software agents sitting inside a server. We look at organoids, for example, neural organoids sitting in a petri dish, and it's very common to say those things are not embodied. They're not embodied because you don't see them rolling around in three-dimensional space doing things the way that we do. But it's important to understand that this perception, action, perception, prediction, memory, action loop happens in all kinds of other spaces. Biology is great at deploying it in other spaces. Living things navigate high-dimensional space of gene expression. They navigate spaces of physiological states and, of course, anatomical morphospace, which we're going to talk about the most now. So we have to be very careful about embodiment. Just because it doesn't have legs or wheels, that does not mean it isn't doing the critical thing for intelligence, which is this kind of perception action loop with all the stuff that can be packed in between. We are not good at recognizing or visualizing these things in other spaces. And this is the sort of thing we are all made of. So this is one cell. This one's a free-living organism called a lacrimary, but you get the idea. There's no brain, there's no nervous system. The whole thing is very competent in its local, tiny little agendas that it has around its own scale. And not only are the cells very competent, but actually the material inside the cells, so the chemical networks themselves, are capable of several different kinds of learning. If you test molecular network models, you will find out that as a free gift from the mathematics, in other words, you don't need evolution for this, although evolution certainly optimizes the heck out of it. But the material itself already is capable of habituation, sensitization, and associative conditioning. So in other words, Pavlovian learning. It can also count to small numbers and so on. So and you can see all that here. And then we are trying to take advantage of this in the lab, of course, for applications like drug conditioning and things like that. Not only can these chemical networks learn, but actually, we found, this is a recent result. A paper just came out a week. Our paper came out a week ago. We showed that the process of learning, so stimulating various components of this chemical network to cause learning, actually increases causal emergence in the network. It becomes more than the sum of its parts. The more that it's trained. It's a very interesting loop. And the thing is that this type of multi-scale scenario where the material has protocognitive properties, the cells that comprise it also do, it goes kind of all the way up. So, Descartes liked the pineal gland in the brain because he thought that, because there's only one of them, and he thought that suited kind of the unified human experience. Fine, but he didn't have good microscopy available. If he did, he would have seen that there isn't one of anything. We are all collective intelligences, in the sense that if you look into the brain, of course, you see all kinds of stuff. If you look into the pineal gland, this is what it's made of, tons of cells. And inside each one of those cells is all of this stuff, and you can keep going. So all intelligence, I would argue, is collective intelligence. We are all made of parts, and the goal of significant agents is to bend their components to their will, to distort the option space for their parts, such that the things they're doing actually feed into goals that occur in other spaces. And so I think what's happening in biology is that our bodies are in fact a multi-scale competency architecture, all the way from the molecular networks up, and every level distorts that option space and produces various attractors and so on, such that the parts do what they do, but as a result, they are actually implementing goals in other spaces of which they are completely unaware. So I want to show you a couple of biological examples of this. So this is that axolotl, amazing little amphibian, highly regenerative. What it can do is if this leg is amputated at any position along the way, the cells very quickly realize that they've been deviated from the correct state in anatomical space. They try to get back by regrowing the limb, and then they stop. This is the most amazing aspect of regeneration, is that it knows when to stop. When does it stop? It stops when the correct limb has been completed. In other words, it's an anatomical homeostatic loop that traverses that morphospace, and it can tell when it's within acceptable error of the starting condition, and then it stops. But this is not about repairing damage. The real story here is not that at all. The real story is about top-down control, because if you were to take a tail and graft it to the side, to the flank of this animal, what you would find is that over some weeks' time, the tail would actually turn into a limb. Not only does the tail remodel into a limb, but these cells up here, which are tail tip cells sitting at the end of a tail, so locally they're fine. There's nothing wrong. There's no damage up here. There's nothing wrong with them, but they turn into fingers. And what's happening here is that all the molecular biology that's needed to respecify these cells into finger cells, right? So all the gene expression changes and the cell movements and all of that stuff is being driven by a high-level goal-seeking process. The large-scale homeostatic loop can tell that it's been deviated from a scenario where what you have on your flank are limbs, not tails. And those control signals propagate all the way down to the cellular and molecular level to make this happen. Now notice that this is a key property of cognitive systems. When you wake up in the morning and you have very abstract social goals, financial goals, whatever you have, those are very abstract things. But in order for you to get up and walk and execute on those goals have to be transduced to make the calcium ions dance across your membranes. right? So the actual chemistry of the potassium channels and all of that stuff has to be harnessed by these higher level loops in order for you to do goal directed behavior. And that is what that multi-scale architecture enables to happen, is that every level is able to bend the option space of the level below, and thus it transduces increasingly more abstract goals. Individual cells have no idea what a finger is or how many fingers you're supposed to have, but the collective absolutely does, and you can see that in these examples. So now, how does it work? Again, I could do hours on this, but just to give you a very brief introduction, about 20 years ago, we made the first tools to read and write the electrical states inside of tissue. Why? Because if we wanted to understand goal-directed behavior and top-down causation, we took inspiration from the brain, which is the one uncontroversial example where we know that happens. So we know we have high-level goals, and we know we can work to solve problems and so on. And the cognitive glue that allows this to happen in the brain, meaning why do we know things that our neurons don't know? Why are we pursuing goals that our neurons don't have? Is because there's this kind of set of policies and mechanisms that bind those competent subunits to common purpose, they align them in various problem spaces. And that cognitive glue is electrophysiology. It's the electrical signaling in the brain. Well, it turns out that the ability of electrical networks to do that was noticed by evolution a really long time ago. It's ancient, long before brains and nerves and muscle and so on evolved. All cells, starting with bacterial biofilms, were using electrical networks to integrate information. And I will just point out a couple of examples. So this is time-lapse of an early frog embryo putting its face together. And if we read the bioelectrical signals using these voltage-sensitive fluorescent dyes, what we see are patterns such as this that are basically presaging the future of what this thing is going to do. This is what it's building towards. All the gene expression and anatomical activity is going to arise towards putting an eye here, putting the mouth here, putting the placodes out here. So this is a pattern memory that the cells are going to implement. And if we change this pattern, they implement something else. Of course, this is a nice multi-scale phenomenon. Not only each one of these circles is a separate embryo. So when we poke this guy in the middle here, all these others find out about it. Not only is bioelectricity the cognitive glue within a single organism, it actually has a similar function across organisms because groups can do things that individuals can't do. For example, resist teratogens and so on. So you can see it's a very dynamic kind of computational network, working slower than the brain, but same idea. So having discovered these things, we developed methods to actually write information back into the cells. No waves, no frequencies, no electrodes, magnets, nothing like that. We take all our tools from neuroscience because I think fundamentally neuroscience is not about neurons at all. It's about this cognitive glue property, and so we use the same tools, and we basically hack the same bioelectrical interface that the cells are using to control each other. And that allows us to do things like this. We can inject into the early frog embryo RNA expressing specific potassium channels. For example, they establish a voltage gradient. That voltage gradient is interpreted by the surrounding cells as make an eye, and we can make other organs too. But here it is. So this gut tissue here was told to make an eye. It did. That eye has the same lens, retina, optic nerve, all the same stuff. So these patterns that exist in this, that are stably kept in this electrical medium, these pattern memories, literally, are used to guide the activity of the system at an organ level. We're not telling the eye we're not telling it how to make an eye. We have no idea how to make an eye. It's a very complex organ, but we don't need to. The system knows how to propagate high-level prompts down into the molecular pathways. And so not only will it make an eye from this very simple prompt, but also we can take advantage of all the other competencies of the tissue, such as if we only inject a few cells, so the blue ones are the ones we injected, what they will do is automatically recruit a bunch of their neighbors. You see all this other stuff out here wasn't injected by us at all. These cells realize there's not enough of them to make an eye, so they recruit their neighbors to complete the task, much like other collective intelligences do, such as ants and termites and so on. So this material has all sorts of competencies, and they're all organized around being able to minimize error towards specific goals and taking goal state information from higher levels. right? That is the architecture that's happening here, and that's the problem decomposition that the material natively does. So I won't take the time to talk about all the biomedical stuff, but basically, whereas all of today's biomedicine basically focuses on these kind of bottom-up interventions, we have a whole sort of a panoply of top-down strategies that are allowing us to do some pretty wild things in regenerative medicine, birth defects, cancer suppression, aging, and so on. And so most of our lab is focused on that stuff, and the development of AI-based tools that could enable us to talk literally, to communicate with the various levels, not just the molecular level, which is what all drug discovery is sort of focused on, or genomic editing or those kinds of things, but to talk to these higher levels. So that we could have much more efficient communication and take advantage of all the problem solving that they can already do. And the idea is that biology itself, so my claim is that biology itself tames this complexity by a multiscale architecture of agential systems with agendas, systems with learning capacity, with problem-solving competencies. The price you pay for that is occasional defections. That's cancer. Another price we pay for that is hackability. So there are all kinds of parasites and other things that can hack it. But the big win there is that no system has to micromanage all the way down. They are all basically just hacking each other at very high levels of with prompts, because the material they're talking to is itself a kind of cognitive agent. And so that's what I think is happening in the biology. Now I want to say a couple things about why this actually works and where did this all come from. The thing that drives all this is commitment to an unreliable substrate. Whereas in computing today, we mostly we have error correction algorithms, and we have abstraction layers, and we have all these things, to try to make the data stay still. In other words, the material, you have to be able to trust your material. If you're working at one level, you cannot be worried that the silicon is floating, the numbers are floating off because your silicon is doing something weird. Biology is exactly the opposite, because biology operates with a very unreliable substrate. And so I'll just give you a simple example. That started me on the road to thinking about all this, which is that what caterpillars, these soft-bodied kinds of creatures, have a very particular brain for crawling around on leaves and eating them. They turn into this. They turn into a butterfly. This is a hard-bodied creature, completely different controller needed to operate this. Lives in the three-dimensional world now, doesn't care about leaves at all. It likes nectar. And the brain is quite different. And so what happens during this metamorphosis process is that basically the brain is dissolved, most of the cells are killed, most of the connections are broken, and then you build a new brain. The remarkable thing about this is that memories that are formed in the caterpillar, so if you train the caterpillar, you find out that the butterfly still remembers the original information. That's wild enough that you can keep information while the substrate itself is being torn to shreds. That's already interesting. But the deeper issue here is that the actual memories of the caterpillar are of no use to the butterfly. In other words, specific actions of the controller for a soft-bodied creature that saw stimulus and needed to go eat the leaves, that doesn't work at all for the butterfly. The butterfly doesn't care about leaves. It doesn't move the same way that the caterpillar did. If you're going to survive, if you're going to persist as a memory across this refactoring, you need to be remapped. The actual the fidelity of the information doesn't help you here. The saliency of the information does. In other words, you need to remap it onto a completely new architecture, new effectors, new sensors, new kind of problem space. In fact, a higher dimensional problem space. So all of this is described here. But that just started me thinking about the importance of first of all, the importance of interpreting the information you have. So the information, whatever molecular information it gets from the caterpillar, it cannot use it directly. It has to interpret it. And while this is pretty drastic, right? So we do a little bit of this, puberty and things like that. But it's not as drastic as this. But actually, all cognitive agents face the same paradox. Here's the problem. At any given time step, you do not have access to the past. What you have access to are the memory engrams that the past left in your brain or body. You have the messages from past versions of you. Memories are a kind of communication event on this model. So you get these messages, and now you have to interpret them. Of course, they are sparse because so there's this, like, I'm sure you'll recognize this architecture. There's this encoding event where you throw out a lot of the extra, kind of regularities and so on, and you squeeze it down into this like very tiny n-gram. But then you've thrown away all this information. You have to reinflate it again. You have to interpret it. So we are constantly, in fact, one could say that what we are as cognitive agents are a continuously evolving, self-telling story that attempts to make a coherent, adaptive model of itself and the outside world based on the information that it received. Not sticking to it mechanically, but actually telling the best story it can, because this stuff is algorithmic. So compressing rich past experiences into a thin representation, that's algorithmic and deductive. But this part is creative. You've lost information. You can't possibly know exactly what happened or how past you interpreted these messages. And so what that means, and this is true not only cognitively, it's true anatomically. The reason that all this plasticity works in the body is that the genetic information that you get from past lineages is just a prompt. It's just a message that you can interpret in different ways, and that's the collective intelligence of the body. The goal is to interpret all that information to do something coherent, and this gives rise to all kinds of amazing competencies. For example, this is a cross-section through a kidney tubule of a newt. You'll notice 8 to 10 cells normally build this. You can make newts with much larger cells by giving them higher copy numbers of their genomes. So they're called polyploid newts. And if you do that, the newt is still the same size. How is that possible? Well, you find out that fewer cells make the same structure. And then if you go really crazy and you make very high level polyploid newts, the cells get so gigantic, now just one cell wraps around itself to give you the exact same structure. Now this is remarkable. This is for two reasons. First of all, this is a different molecular mechanism. So this is straight up what people measure on IQ tests. You're given a set of objects and you're asked to solve a problem. How do you use, here are 3 things. How do you use this to solve a particular problem? So this system has a genome. That genome gives lots of affordances. There are ways for cells to communicate. There are ways to bend themselves. You have to use the right tools to solve a problem in novel circumstances. So now think about what it's like being a creature coming into this world. You can't rely on how many copies of your genetic material you're going to have. I mean, never mind the uncertain environment. You can't even trust your own parts. You don't know how much what your genetics are going to be. You don't know how many cells you're going to have or how big your cells are going to be. You have to try to solve the problem in novel ways, and this is why all this plasticity works. The reason those tadpoles work with eyes on their tails, I think, is because they never knew that the eyes were going to be in the front in the first place. They could never count on that. If they are, great, that's a default outcome, but no one can count on that because you know that things will change. You will get mutated on the lineage scale, the environment changes, your own parts change. It's kind of beginner's mind. You have to take what you have and somehow do something useful with it. By the way, this is Bongard's paper in 2006 on robots that didn't know what their shape was really prescient in all of this. And so the deal is that what's happening here is that biology is dealing with a very unreliable medium. You never know how many copies of anything you have or whether your proteins are getting degraded or what. You can't take it literally. But what you can do is develop good skills to interpret it in a useful fashion. That's why this kind of cognitive bottleneck, this autoencoder kind of thing, is exactly what organisms do. They don't make other organisms, they make eggs. And it's up to the cells to interpret the information that was in the egg. And if you actually do evolutionary, I don't have time to go into this, but if you actually look at how evolution works on agential materials, you find something really interesting. You find that because that competency, that problem-solving competency of the material makes it really hard to evaluate genomes, because genomes aren't what come up for selection. What comes up is the actual phenotype, the results. What evolution ends up doing is spending much more of its time cranking improvements in the interpretation machinery. That a confabulatory ability to tell novel stories using whatever information you got with no allegiance to how it was interpreted in the past or what was happening before is what evolution really cranks on. So you end up with this feedback loop, this positive feedback loop between this kind of creative interpretation and the intelligence of the system in solving different kinds of problems. So at this last bit here, I just want to say a couple of things about what I think this all means. You know, all that plasticity makes life incredibly interoperable, and that means that pretty soon we are going to be faced with not just this spectrum where, you know, we all came from a single cell, both evolutionarily and developmentally, but there's another spectrum, which is technological and biological change. So this quote-unquote human that features so prominently in stories of philosophy of mind and so on, is actually, there's kind of a spectrum here that's not easily dealt with by our ancient binary terminology. And we are going to have all kinds of interesting creatures, both you know, made of all sorts of different combinations of evolved and engineered material. And so now we have to ask the following question: if you're dealing with a being that is nowhere on the tree of life with you, meaning they did not come up through the same evolutionary process, can we guess what their competencies are going to be? What are their goals? What are their preferences? What are their competencies? You know, in standard biology here, this morphospace of these planarian heads, there are specific attractors that we can chase these tissues into, and people will say, well, of course, evolution formed these, right? That's what evolution does. But when you're dealing with agents that don't have that simple backstory, what actually happens? And so this is going to be the weirdest part of the talk, and you know, here's where I'm going to speculate as to what I think is happening here. Biologists, as explanations, biologists like two things. They like stories about physics, and they like stories about genetics. In other words, they like explanations for a certain phenomenon which can be cashed out in terms of the necessity of physics and the historical contingency of its environment, you know, and things that have happened in the environment. But we already know that that's not sufficient, and that physics isn't closed like that. And just as a simple example, cicadas come out at 13 years and 17 years. Why? If you're a biologist, you want to understand why. Well, it's because they're trying to time it to periods that are hard for predators to match, and so 13 and 17 are good because they're prime numbers. And you say, okay, so if you keep trying to pursue the chain of explanation, the next question is, okay, why don't 13 and 17 have factors? Now you've left the physical world. Now you're in the land of mathematics. There is nothing in physics that will answer that question for you. There's nothing in evolution that will answer that question for you. You have already, if you want to understand what cicadas are doing, you have to eventually cross over and start looking at mathematical patterns and however why questions are handled in mathematics. So the reality is that genetics and environment are not the only place that complex patterns come from. So this is, I could have given many examples. This is just one of my favorites because I think it looks cool and it's kind of organic. All these things kind of look biological a little bit. This is called the Halley plot, and it is what happens when you map the behavior of this complex number function. It's very simple, this little tiny seed. Talk about small engrams that can be reinflated into complex patterns, right? I mean, look at this. The compression is insane. And if you plot this in a particular way, you get this amazing structure. And the answer to why does the structure look the way it looks has nothing to do with physics. There's nothing you can do in the physical world to change what it is. There's no history behind it. It is a fact of mathematics. Math is full of these things. And then you know, this is just a video I made by iterating this in various ways. So there's very rich structure buried in the rules of mathematics, and biology exploits these things like crazy. These are amazing free lunches that biology uses all the time. And so in the last few minutes, I just want to show you some biological life forms that have this kind of character where you can't really use selection history to explain them. If I showed you this little creature, you might think it's something we got off the bottom of a pond somewhere, like a primitive organism. So actually, if you were to sequence it, you would find out that it's 100% Homo sapiens. So adult human cells extracted from the trachea of patients doing biopsies under a particular protocol that we developed form this little self-motile creature. It looks and acts nothing like any stage of human development. It has some amazing properties. It has all kinds of behaviors. It has about 9,000 genes expressed differently than the tissue did when it was in the patient. So about half the genome is completely remodeled here. Remember, no changes to the genome, no synthetic biology circuits, no scaffolds or weird drugs. All we did was liberate those cells from their environment and let them reboot their multicellularity. This stuff out here is human neurons, and what we did was make a big scratch through it like this. And these anthrobots, which is what we call these little creatures, they land in this kind of like superbot cluster, and they actually start healing across the gap. Lift them up 4 days later. This is what you see. Why are they doing this? We did not teach them to do this. This is spontaneously what they do. This is the first thing we checked. So probably they do 1,000 other things we don't know, but they have this amazing ability. And who would have thought that your tracheal cells that sit there quietly dealing with mucus and air particles have the ability to form a little creature that can assemble and do this kind of thing. These are xenobots, so it's a similar idea made with frog cells. They have an amazing capacity. They have many, but this one thing I'll show you is called kinematic replication. So if you give them a bunch of loose epithelial cells as a material, they will collect it into piles. The piles mature and become the next generation of xenobots, and then guess what they do? They go on and they make the next generation and the next again. We didn't teach them to do this. So the amazing thing with all of this is that this again goes back to the plasticity of the material, and this idea that okay, yeah, the genome learned to do this over many generations. This is the standard developmental sequence. Here are the tadpoles, but it also can do this. So this is an 84-day-old xenobot, and many other kinds of things like kinematic replication. There's never been selection to be a good xenobot or a good anthrobot. There's never been any anthrobots or xenobots. Where do their capacities come from? You cannot tell a story of selection that matches it. And if you say that these are emergent from the things that the cells did do in the environment, what you're basically giving up is any degree of specificity between the final outcome and the history of selection that they've had, which is basically what evolution was supposed to do. It was supposed to explain the specificity and give you a causal story, right? So there's some very interesting features here. Like we know when we paid for the computation to do all of this. That was during the evolution of the frog. When were all the computations done to get this going? There was no history of this. Where did this come from? This is very interesting, and the idea of these kinds of things that have no obvious history of either learning or selection is I think really demands attention. And there is a precedent for this, and the precedent now biologists hate this idea of the physical world not being closed and of needing to go outside. They like a nice sparse ontology. But mathematicians are fine with this. There are many Platonist mathematicians who believe that there are patterns that they are discovering, not creating, discovering, and that the order that the space containing these patterns is a structured, ordered space which they navigate through their research. Actually, also which I don't have time to talk about now, but this is actually starting to crop up in machine learning as well, which they call the Platonic space hypothesis. So what I'm gonna basically just say is this. People who like their sparse physicalist ontology, which basically will lean on emergence and this idea that, look, there are just some facts that hold in the physical world. You have a system with simple rules. It can do complex things, and that's it. There's just these surprises. These things are emergent, and I don't like that view because I think it's Mysterian. I think that it relegates these things to surprise. Basically, emergence then is anything we find surprising. You write them down in your big book of emergence, and that's the end of it. I like the more positive view of the mathematicians where they're not haphazard things that just happen to hold in the physical world. There is an ordered, structured space that we can rationally study. How do we study them? By producing pointers into that space. And so everything that we make, cells, embryos, biobots, AIs, hybrids, cyborgs, all this stuff, all of them are pointers into that space. We can use them as exploration devices or interfaces to try to understand the mapping between the thing that you built and the patterns that are ingressing through this space. I would like to hope that I can't prove it. It's a metaphysical assumption that the space is understandable. It's not just a random grab bag of stuff. And so What I do think is important, and this is if I haven't lost you yet with the previous thing, this it gets even weirder here, is that I think it actually is not just for the biologicals. It's not just the complex living things that get to have the benefit of these ingressions, these free lunches from mathematics. It doesn't take much at all. And if you're interested, this is one example, and I chose sorting algorithms on purpose because they are so minimal and so deterministic and so obvious that to kind of maximize the shock value of this. Take a look at this paper. What we found is that even the simple things like bubble sort have unexpected side quests, the things they're doing that are literally not in the algorithm. They're nowhere in the algorithm. And so that means like all the rest of us biologicals, there are things we have to do. We are forced by the physics and by the genetics of our world to do certain things and are limited in certain ways. But for the time that they're alive and operational, before they get ground into dust by the physics of their world, there are other things that they do that are nowhere in the algorithm. And for this reason, I think that, and by the way, the things that they're doing would be things that behavioral scientists would recognize immediately. If you didn't tell them that this came from an algorithm, they would immediately say, "Oh yes, this is something we see all the time in our among the sort of set of behaviors that we've seen." These are behavioral competencies that arise that cannot be explained simply by looking at the algorithm. They're not inconsistent with the laws of physics any more than our cognition is, but they're not prescribed by it. And I think it's very important to understand that even if these kinds of things can be, if even dumb bubble sort can be an interface to interesting behavioral propensities from this latent space, then what do we say about the Internet of Things and language models and all this other complicated stuff? I mean, I get really frustrated when people say, "Well, I write these language models. It's just linear algebra. There's nothing in there." Right? If we never knew after 60 years or however long of working with bubble sort, if nobody had noticed it doing these things, what else do we not understand? And so, I want to make a claim here, which is that our formal models do not circumscribe the cognitive aspects of the objects that they're supposed to understand. No more for us. So for the exact same reason that that the rules of biochemistry do not fully describe what our minds are capable of, the algorithms and the stories of materials and everything else that we try to use to understand simple machines, quote unquote, do not fully circumscribe what they're doing either. And so my point is that there are limitations, and these are limitations of our formal models. They are not limitations of the actual thing. And there are massive surprises, certainly on the biological end, but it goes all the way down. And my point is not to say that living things are machines in the sense that, yes, we're all on the same spectrum, but not because you can use simple machine metaphors to understand life. Precisely the opposite, because even quote-unquote machines are doing things that our simple mechanical formal models do not capture. The magic goes all the way down, I would say. And so I'll just I'm gonna stop here in a minute to just point out that we are soon going to be living in a very wild world full of all kinds of different embodied minds. Right, all kinds of combinations of software, synthetic materials, evolved materials, and all of the ingressions from this latent space that they allow. We are very poor at predicting what is going to come through, what the behaviors are going to be, and so on. And we need to get a lot better at this so that we can have an ethical symbiosis with all these things and for different types of human flourishing that I think we're all interested in. So the bottom line is simply this, that I think we need to really do bio-inspired computing. We need to focus on unreliable agential materials, a flexible plastic cognitive light cone, complexity that is dealt with by top-down control. And I didn't even get to mention this, but this thing that Josh Bongard and I are developing called polycomputing. Embodiments are much broader than we think they are. I'll just come out and say it, as unpopular as this is. I think the physicalist assumptions hold us back. They hold us back in biology, cognitive science, and computer science. I don't think emergence and complexity are sufficient for what we need to do. And I don't believe we make intelligences, either biological or technological. We make interfaces that are exposing us to an amazingly interesting latent space that we need to understand. And I think that's our research agenda. I'm going to stop here just to say that there's a whole bunch of papers. I'm happy to send these to anybody who's interested. This is all the people that I need to thank, the postdocs and the students who did some of the work that I showed you today. Are lots of amazing collaborators, our funders. Disclosure, there are 3 companies that have spun out of our lab that fund some of the work. So these are the commercial entities. And then, the model systems do all the heavy lifting. So I always thank them. So I thank you for listening.